Hello friends, today I will tell you about designer nucleases for genome editing in plants, different approaches and applications. Same techniques are using nowadays in animals also, but today I will focus only on plants. So let's start. These are the different methods by which we can modulate gene expression. So there are various methods for modulation of gene expression at various levels. If your approach is stable and site-specific integration, this can be done easily by zinc fingers or zinc finger nucleases or TALENs. At DNA level, we can use zinc finger nucleases, TALENs or CRISPR-Cas system, siRNA at RNA level, and we can also inhibit protein product by producing monoclonal antibodies, or we can also design small peptide aptamers against a specific protein. Firstly, talk about what is genome editing. Genome editing is the use of engineered nuclease to introduce, insert, or replace a gene at a targeted genomic location. Such engineered nucleases typically comprise two elements. First, a DNA cleavage module, which is generally a nuclease, which can generate a double-stranded break anywhere in the genome and thus inducing cell repair process. And the other part will be a sequence-specific binding domain that can customize to recognize any sequence. This part can be zinc finger protein or tall protein or CRISPR guide RNA. This scheme here give you an idea about possibilities to manipulate different elements contributing to the expression of a gene by using genome editing. So what can you do with genome editing? You can introduce point mutation at exons. You can disrupt a gene function by deleting a small portion or a large segment of a gene, and you can also insert LOX P site for site-specific recombination process, or you can integrate an expression cassette anywhere in the genome. A nuclease can cut a double-stranded DNA and create a double-stranded break anywhere in the genome, and this can increase mutagenesis frequency by more than 1000-fold. There are two types of repair process that can occur after double-stranded break. Without a homology or an external donor DNA, the broken ends can be rejoined, and this is called non-homologous end joining or NHEJ, and this often accompanied by a mutation which can cause deletion of a functional element of a gene, and in presence of donor DNA, insertion or replacement of a gene can take place via homology dependent repair or homology directed repair process, or HDR, also known as HDR. The ratio of non-homologous end joining and homology directed repair process depend upon the individual experimental setup, like cell type and amount of donor DNA. This is zinc finger protein. Zinc finger proteins are most common family of transcription factors that regulate transcription in eukaryotes. Each zinc finger protein consists of approximately 30 amino acids which folded into beta beta alpha structure. In this protein, zinc coordinate with conserved cysteine and histidine residue, and each zinc finger motif bind a triplet within a DNA structure. Zinc finger motif for all 64 possible triplets are available, so we can target any DNA sequence in the genome. The first zinc finger nucleases were created as chimeric restriction endonuclease and were shown to have in vitro activity. Kim et al. Designed artificial zinc finger nuclease by fusing different zinc finger domains to the fork-first nuclease. Fork-first is a non-specific nuclease. It can cut to any targeted location in the genome. So this was done for the first time in 1996 and published as a proof of concept by Kim et al. Zinc finger nucleases, once bound to the targeted loci, dimerize in the cell, and this induces first nuclease to create double-stranded break. This activates cell repair process as discussed in previous slide, and this result in gene knockout, mutation, and insertion of a DNA fragment in the genome. In maize, the herbicide-resistant gene PAT was inserted at the IPK1 gene locus, causing simultaneous disruption of IPK1, which encodes inositol 1,3,4,5,6 penta kiss phosphate 2 kinase enzyme that involved in phytate biosynthesis in maize seeds. So when this gene is knocked down, phytate level in the seed is reduced. Phytate accounts for 75% of anti-nutritional phosphorus and also contribute to environmental pollution when corn seeds are released in the environment as waste. So the objective of this study was twofold. First, to reduce IPK activity in maize, and second, to impart herbicide resistance to the corn plant. And these are some successful examples of genome editing in plants by using zinc finger nucleases. Friends, second type of artificial nuclease is TALENs, stand for Transcription Activator like Effector nucleases. In 2009, transcription activator like effector proteins or TALs have been discovered which use simple model for DNA recognition. TALs are naturally occurring proteins from the plant's pathogenic bacteria and contain DNA binding repeats. Each TAL protein recognizes a single nucleotide in the DNA. A tall protein contains a nuclear localization sequence, DNA binding domain, and transcription activator domain. Each DNA binding domain contains tandem repeats of 33 to 35 amino acid motif, and each motif of tall protein recognizes a single nucleotide or base pair through 2RBD residue, means divariant residue. Talls are typically fused with first nuclease and used for targeted genome editing in plants and animals. Talls can be fused to transcription activators to trigger any gene in plants or animals, or can be in bacteria. So once TALENs create double-stranded break in the genome, this break induces cell repair process which results in gene knockout, gene modification, or insertion of a DNA fragment anywhere at the targeted loci in the genome. Friends, this is a successful example of genome editing in wheat using TALENs. This work was conducted by Wang et al. And published in Nature Biotechnology in 2014. In wheat, powdery mildew disease is caused by Blummeria graminestriaceae (BGT) fungus, and this is one of the most destructive plant pathogens worldwide. Two wheat varieties, Genome and Bob White, were selected for this experiment. Tolerance induced loss of function of MLO alleles generate broad spectrum and durable resistance to the BGT fungus. MLO loci were selected for genome targeting experiment because this loci encode protein that were shown to repress plant defense mechanism against fungal pathogen. So in this study, tolerance-induced mutation of all the three TAMLO homologs in wheat confers heritable broad-spectrum resistance to the powdery mildew disease. These are some other successful examples of genome editing in plants using transcription activator like Effector nucleases. Now coming to CRISPR-Cas system. CRISPR-Cas system was discovered in E. coli by Ishino. CRISPR stands for clustered regularly interspaced short palindromic repeats. CRISPR system is a bacterial adaptive immune system to defend against invading viruses. CRISPR RNA and transactivating CRISPR RNA form complex with invading virus genome, and Cas9, which is an endonuclease, cut the genetic material of virus and protect bacteria against virus attack. Palm sequence must be present near target site for Cas9 action. So in modified system, we can change CRISPR RNA and transactivating CRISPR RNA with a chimeric short guide RNA to create targeted break anywhere in the genome. This figure shows how CRISPR-Cas system works. Single guide RNA with Cas9 search or scan for target DNA sequence, and wherever target sequence found, Cas9 create double-stranded break at that particular location in the genome, and this double-stranded break result in gene knockout or integration of desired DNA at selected location in the genome, similar to Zinc-finger nucleases and TALENs. This is an example of CRISPR-Cas9 induced genome editing in rice plant. In this study, the role of sugar transporter OS sweet 11 gene was investigated by Ma et al. In 2017 during the early stage of rice caryopsis development. For this study, OS sweet 11 gene was knocked out using CRISPR-Cas9 system, and in embryo sac of transgenic rice plant, sucrose concentration was decreased, which led to defective grain filling compared with wild type plant. So this study showed OS sweet 11 gene expressed strongly in developing caryopsis of rice plant. These are other examples of CRISPR-Cas9 system induced genome editing in different plants. Here, this table is showing the differences between Zinc-finger nucleases, transcription activator like Effector nucleases, and CRISPR-Cas9 system. So the nuclease type is fork first in both Zinc-finger nucleases and transcription activator like Effector nucleases, but it is Cas9 in CRISPR-Cas9 system, and DNA binding domain is formed by zinc fingers in zinc finger nuclease and tall proteins in transcription activator like effector nucleases, but in CRISPR-Cas9 system, DNA binding domain is formed by single guide RNA. The type of nucleases: zinc finger nucleases is a fusion protein and TALENs also a fusion protein, but CRISPR-Cas9 system is a combination of protein and RNA. In zinc finger nucleases, binding site is formed by two sites, 15 to 18 nucleotide long, and in TALENs, DNA binding site is also formed by two sites, which are approx 13 base pair long, but in CRISPR-Cas9 system, only one site is used for DNA binding by CRISPR-Cas system, and this DNA binding site can be 18 to 20 base pair long, and in addition 3 base pair NGG sequence, which is palm sequence, is also required. Friends, this is my paper on designer nucleases for genome editing in plants. You can download and read this paper. This is freely accessible. Friends, thanks for watching my video. If you like this video, please subscribe my channel and share my video. Thank you.